All right, welcome back to the deep dive. Today, we're taking a closer look, a deep dive, into, a pretty fundamental area of clinical lab science. We're talking about carbs.
Yeah, carbs, carbohydrates.
Carbohydrates, exactly. But we're not just going to skim the surface here. We're going to really put on our, our lab coats, our medical laboratory scientist hats, and really dive into how these molecules, these biochemicals, really impact the work you do every day.
Absolutely. This isn't just your biochemistry 101 review. This is really connecting the dots between the chemistry, the fundamental chemistry of carbohydrates, right? The metabolic pathways that they're fueling, how those tests that we run, the principles behind them, how we interpret the results, and then, of course, tying it all back to disease. We've looked at a lot of different sources for this one. Everything from the general chemistry of carbohydrates and their properties to, really the complexities of how we use them and store them, how things go wrong, the theory behind all the tests we run, and all those little crucial details that can make or break getting the right results.
Yeah, that's really what we're here for, is to extract the most crucial knowledge, the stuff that really matters for your work in the lab and really making some connections that you might not always make. So ready to jump in?
Let's do it.
Okay, so let's start with those biochemical foundations. We know carbohydrates are our body's primary fuel. And glucose is the undisputed champion in that arena, right?
Absolutely. Think of glucose as the, energy currency of the body. And what's so fascinating is, how that currency is managed, right?
Right.
This intricate network of metabolic pathways. Each one has its own specific role, location, the whole 9 yards. So why don't we kick things off with glycolysis that's occurring in the cytoplasm of the cell. What's going on there?
So glycolysis takes glucose, breaks it down into pyruvate, and in the process, we get a little bit of ATP, that immediate cellular energy, and NADH, another important energy-carrying molecule. Now, the fate of pyruvate, that's an interesting one, isn't it?
It is. It's kind of a fork in the road, right? Determined by oxygen.
Right.
Oxygen's around. Pyruvate can be further oxidized, generate more energy. But if oxygen's limited, think intense exercise, that pyruvate gets shunted to lactate. That anaerobic pathway keeps things chugging along.
Right, keeping that ATP production going.
At least some ATP. Now, what about gluconeogenesis?
Oh, gluconeogenesis. That's like our body's backup generator, right? Making new glucose when supplies are low.
Precisely. Primarily in the liver. Gluconeogenesis is humming along when we're fasting or starving, pulling in those non-carbohydrate sources, things like lactate, glycerol, amino acids.
Right.
It's the key, the absolute key to maintaining stable blood glucose levels, between meals. And that's what you're measuring in the lab all the time, right?
Exactly. That's what we see. So when we eat and that blood glucose rises, that extra glucose isn't just wasted. The body has a plan, a storage plan.
Exactly. And that's glycogenesis.
Right.
Liver, muscles, they become our warehouses. They're converting that excess glucose to glycogen, that more complex carbohydrate, for later use.
Yeah.
Like our readily accessible glucose reserve.
Right, exactly. And when those energy demands increase or that blood glucose starts to fall,
The glycogenolysis kicks in. It's like mobilizing reserve.
breaking down that glycogen, releasing glucose right back into the bloodstream. It's fascinating.
Right.
Yeah. Now, we can't forget about the pentose phosphate pathway,
The PPP.
the PPP.
Yeah.
Maybe not directly involved in ATP production, but it's incredibly important.
Oh, absolutely. It might not be the star of the energy show, right?
Right.
But it has some very important roles. Think NADPH production.
Yeah.
That's crucial for building things like fatty acids, steroids. It's our cellular defense against, oxidative damage, regenerating those antioxidants.
Wow.
And it makes ribose 5-phosphate.
Right.
Key component for making nucleotides, the building blocks of DNA and RNA. So it's involved in all sorts of fundamental processes beyond just, keeping the lights on.
It's amazing how interconnected all these pathways are and what's orchestrating this whole intricate system.
Hormones. Insulin, glucagon, epinephrine, cortisol, they're the conductors, right?
Right.
Signaling these pathways to speed up or slow down based on the body's needs.
Right.
You can think of insulin as like, the storage hormone. It's telling the cells, hey, plenty of glucose, let's store some up. Glucagon's on the other side saying, we need more fuel, let's break down those stores.
And release.
Exactly, but it's even more finely tuned than that, right? It's not just, on or off. These hormones, along with allosteric effectors, things like ATP and AMP, which really reflect the cell's energy status, they're constantly providing feedback, right? Adjusting the activity of those enzymes to maintain that critical glucose homeostasis.
Right.
The stable blood glucose levels.
Yeah.
Like a thermostat for blood sugar.
Perfect.
And that's what you're measuring, day in and day out.
Exactly. So we've got this beautifully balanced system. But what happens when the balance is disrupted? And that's where, we see those physiological states and disease correlations that are really at the heart of what we do.
Exactly. In a healthy state, we've got normal glycemia, right?
Right.
Those normal fasting and postprandial glucose ranges. That's metabolic function working as it should, maintained by that hormonal symphony we just talked about. But, of course, things can go wrong, right?
Of course.
And that's when we see those abnormal states. And I think the most common one that we deal with is hyperglycemia.
Right. Elevated blood glucose.
Exactly. And the causes, they're diverse. Diabetes mellitus is the most common, of course, whether it's type 1 with the autoimmune destruction of those beta cells, type 2 with insulin resistance, gestational diabetes during pregnancy. But there are lots of other conditions that can lead to hyperglycemia too. What's clinically significant is recognizing those complications that are associated with hyperglycemia.
Right.
Both acute hyperglycemia, like DKA, diabetic ketoacidosis, HHS, hyperosmolar, hyperglycemic state, those are the ones that need immediate attention. And then you've got those long-term chronic complications affecting the eyes, the kidneys, nerves, the cardiovascular system.
Right. And then on the other end of the spectrum, we've got hypoglycemia, low blood glucose, which can be just as dangerous, if not more so in some situations.
Absolutely. And again, lots of different causes. It could be excess insulin in a patient with diabetes. It could be liver disease, kidney disease, hormonal deficiencies, even, too much alcohol.
Right.
And as you know, the consequences of severe hypoglycemia, they can be pretty dire. Neurological dysfunction, seizures, coma, and even, permanent brain damage.
We also hear a lot about insulin resistance. So where does that fit in?
Think of insulin resistance as, the body's cells are becoming less responsive to the signals from insulin, right?
Right.
Hey, take up some glucose.
Right.
And to compensate, the pancreas often pumps out even more insulin, leading to hyperinsulinemia. And that insulin resistance, that's a key player in prediabetes, type 2 diabetes, and the whole metabolic syndrome picture.
Right, yeah.
And understanding at the cellular level, it really helps explain why, just increasing the insulin dose in type 2 diabetes, it eventually becomes ineffective.
Yeah.
And you might see that reflected in how physicians order tests as they're trying to adjust treatment strategies. Now, we can't forget about those genetic disorders that directly impact carbohydrate metabolism.
Yeah, there are a number of those conditions.
There are glycogen storage diseases, the GSDs, genetic defects in those enzymes involved in either making or breaking down glycogen.
Right.
Leads to abnormal glycogen accumulation or, not enough glycogen when you need it. Then you've got galactosemia, where there are enzyme deficiencies needed to metabolize galactose. That can cause some serious health problems if it's not caught early.
Yeah. And I recall that with hereditary fructose intolerance, that aldolase B deficiency can lead to that buildup of fructose 1-phosphate, which can be toxic to the liver and then lead to hypoglycemia after you ingest fructose. So early dietary intervention is so important.
It's absolutely critical.
Yeah.
Early diagnosis, often guided by, recognizing patterns in those routine labs or through specialized assays. And then, strict dietary management can make a huge difference. We also see things like pentasuria, typically a benign condition with pentose is being excreted in the urine. And then, of course, we have to really delve into diabetes mellitus. I mean, given how prevalent it is, you see it in the lab all the time.
Yeah, all the time. And it's not a single entity, right? We have type 1, that autoimmune form, type 2, the insulin resistance, and gestational diabetes during pregnancy. And those lab findings, they're really key to diagnosis.
Yeah, absolutely.
We're looking at a fasting plasma glucose of 126 milligrams per deciliter or higher, a 2-hour OGTT result of 200 or higher, an HbA1c of 6.5% or higher, or a random glucose of 200 or higher with symptoms. And then, of course, the presence of ketones is really important, especially in cases of DKA.
Right. Those are the cornerstones of diagnosis. And I think for you in the lab, it's so important to appreciate how those elevated glucose levels, right?
Right.
They correlate with those long-term complications. Yeah. And then how the presence of ketones, that's signifying that acute metabolic crisis of DKA.
Right.
And then you've got prediabetes. It's that, middle ground, right?
Yeah, it's that in-between stage.
Yeah, where those levels are higher than normal, but not quite meeting the criteria for diabetes.
Yeah.
So we've got impaired fasting glucose, IFG, in the 100 to 125 range, impaired glucose tolerance, IGT, with that 2-hour OGTT result between 140 and 199, or an HbA1c between 5.7 and 6.4%.
Right.
But it's significant, right? It means increased risk for developing type 2 and cardiovascular disease down the road.
Right. And gestational diabetes, that, requires its own set of screenings and diagnostic procedures, typically involving that glucose challenge test followed by a full OGTT if that initial screen's positive.
Yeah.
And you'll often see those Carpenter-Kustin criteria being used,
Right.
which have specific glucose cutoffs for fasting, 1 hour, 2 hour, sometimes 3 hour time points.
Exactly. And recognizing those criteria and those risks for both the mother and baby, that's so important. And then we have those acute hyperglycemic emergencies, DKA and HHS.
Yeah.
Both involve very high glucose levels, but there's some key differences in their lab profiles. So DKA, it's driven by lack of insulin, right?
Mm-hmm.
And it typically presents with glucose above 250, very elevated ketones, a low blood pH, so acidosis, low bicarbonate, and a high anion gap,
Right.
reflecting that buildup of acids. HHS, on the other hand, that's more often seen in type 2 diabetes, usually triggered by some illness. And you see those super high glucose levels, often over 600.
Wow.
Osmolality is elevated, but ketones are usually absent or just mildly elevated, and the pH and bicarb are, closer to normal. And of course, both of these are, critical medical situations. You need accurate lab data fast.
And speaking of that anion gap in DKA, when you see that increase in the lab results, what specific electrolyte imbalances are you actually seeing there?
Well, we typically see elevated unmeasured anions
Right.
as the body produces those keto acids.
Mm.
And that leads to a wider gap between the measured cations, like sodium and potassium, and the measured anions, like chloride and bicarb. So it all ties back, right? The biochemistry and those specific analytes you're measuring.
Mm-hmm.
And then you mentioned, galactosemia. Those lab findings are going to include elevated galactose in blood and urine. And the definitive diagnosis often involves demonstrating that specific enzyme deficiency.
Right.
And with hereditary fructose intolerance, you might see hypoglycemia after fructose ingestion, elevated liver enzymes, and then you might see those non-glucose reducing substances in the urine.
Right. So all these different clinical conditions, they've got their own specific fingerprints in the lab. Now, let's shift gears a little bit and talk about how we actually generate these lab results. What are the fundamental principles behind those carbohydrate tests that we run every day?
Well, glucose measurement, that's really the cornerstone, right?
Yeah, I guess.
And the workhorse methods, the ones we rely on the most, are the enzymatic methods.
Right.
So you've got the glucose oxidase method, GOD.
Right.
That involves the oxidation of glucose, producing hydrogen peroxide, which then reacts with a chromogen to give you that colored product.
Right.
And then you measure that with a spectrophotometer.
And that intensity of that color, that's directly proportional to the glucose concentration in the sample.
Exactly. Now, I know you're aware there are some analytical challenges with this method.
Right.
It can be prone to interference from reducing substances,
Yeah.
like ascorbic acid, vitamin C,
Right.
which can lead to falsely low glucose results. So that's something to really keep in mind, especially with certain patient populations.
Mm-hmm.
And then you have the hexokinase method, which is considered to be much more specific,
Right.
often used as a reference method. And this one, it involves the phosphorylation of glucose by the enzyme hexokinase. You get a series of coupled enzymatic reactions that ultimately produce NADH,
Right.
which is measured by its absorbance at a specific wavelength.
And that NADH production, that's what our lab instruments are detecting as a change in absorbance at 340 nanometers, right? And that change is directly proportional to the glucose concentration in the sample. And we can use different specimen types for these enzymatic assays: plasma, serum, even whole blood, especially for those point-of-care tests.
Right.
And of course, each one has its own pros and cons in terms of ease of collection, potential for interference, and cost.
Absolutely. Now let's talk about the glucose tolerance test, the OGTT.
Right, the OGTT.
That's a much more dynamic assessment.
It is. It really is. It's really assessing how efficiently the body is clearing glucose from the bloodstream
Exactly.
over a certain amount of time. So the patient drinks a standardized glucose load, and then we measure those blood glucose levels at specific intervals, giving us a really good picture of insulin secretion and glucose uptake.
Exactly. And it's so valuable for diagnosing not only diabetes, but also gestational diabetes, and for identifying insulin resistance. But that patient prep, it's critical.
It really is. Proper carb loading for a few days before, an overnight fast, avoiding strenuous activity or smoking before and during the test. All of that's essential for reliable results.
Absolutely. And don't forget about medications. Some medications can really impact glucose metabolism, so it's always important to know what the patient's taking. And then the handling of those blood specimens is equally important, typically using those fluoride oxalate tubes
Right.
to inhibit glycolysis, that breakdown of glucose by red blood cells. And those samples need to be processed quickly
Right.
to prevent those falsely low values.
Yeah. Now we've got to talk about glycated hemoglobin, HbA1c. It has really become essential for long-term glucose monitoring. So what's the principle behind this test?
It's reflecting the average glucose over the past 2 to 3 months. That's the lifespan of a red blood cell.
Right.
And it's based on the non-enzymatic glycation of hemoglobin by glucose. So the higher the average glucose, the higher the percentage of hemoglobin that's glycated.
Right. And the beauty of it is that, unlike a single glucose measurement, fasting is not required. And there are several different methods to actually measure HbA1c in the lab.
Yeah, there are a bunch. There's
Yeah.
ion exchange chromatography.
Right.
That separates hemoglobin fractions based on charge differences.
Yeah.
Then you've got affinity chromatography. It uses boronate, which binds to the glycated parts of hemoglobin.
Right.
You've got immunoassays that use antibodies that recognize HbA1c specifically. And then capillary electrophoresis separates them based on their electrophoretic mobility.
Right.
Each method has its own strengths and weaknesses. What's really interesting is how seemingly unrelated conditions, like anemia, can impact this key marker of glucose control.
Right.
So we have to be really, really careful in interpreting the results. And sometimes that means using a specific assay that's less affected by those interferences.
Right. And we always need to be mindful of the potential for interference. We talked about hemoglobin variants, and that can really affect the accuracy of some of the methods. Also, anything that alters red blood cell turnover, like anemia or hemolysis, can impact those results. Even ethnicity, pregnancy, and kidney disease can affect HbA1c levels.
Right.
So using the right testing methodology is critical, especially if we know the patient has a specific hemoglobinopathy.
Absolutely.
Now, urine glucose, it's not as common as it used to be, but it still has its place.
It does.
So the basic principle there is that glucose will only spill into the urine when that blood glucose level exceeds the kidney's ability to reabsorb it. That's the renal threshold.
Exactly.
So it's not quite as sensitive, is it, compared to direct blood glucose measurement?
No, it's much less sensitive. A negative urine glucose doesn't rule out hyperglycemia.
Right.
And a positive result just means that renal threshold has been crossed.
Right. Okay, and ketone measurements, those are critical in the assessment of DKA.
Absolutely. So these tests, they detect the presence of ketone bodies, primarily acetoacetate, acetone, and beta-hydroxybutyrate. And those are produced when the body starts breaking down fat because there's not enough glucose available.
Right. So the traditional nitroprusside-based tests, like the urine ketone dipsticks, they mainly detect acetoacetate and acetone. But in DKA, the main ketone body is beta-hydroxybutyrate.
Right.
So some labs will use those more specific enzymatic assays to directly measure beta-hydroxybutyrate, giving us a more comprehensive assessment.
Exactly. And for those, less common inherited metabolic disorders like galactosemia and hereditary fructose intolerance, there are those specialized enzymatic or chromatographic techniques to measure those sugars, the galactose and the fructose, in blood and urine.
Yeah.
And to also look at the activity of those specific enzymes involved.
Right. So a whole range of tests, each with different biochemical principles. Now, once we have those results, interpretation, that's the crucial step. So what are some of the things we need to keep in mind?
Well, for glucose, fasting, random, postprandial, we interpret those values within those established ranges, normal, pre-diabetic, diabetic.
Right.
And those high levels, that's a big red flag for potential diabetes or just poor glucose control.
Right.
With the OGTT, we're looking at that whole curve over time, right? Comparing it to the diagnostic cutoffs.
Right.
Using those specific criteria. And with HbA1c, we interpret that percentage in terms of long-term control.
Right.
Again, using those established ranges and targets. But we have to remember the limitations of HbA1c in certain cases.
Absolutely.
Like we've talked about. A positive urine glucose, it simply means there's enough hyperglycemia to exceed that renal threshold.
Yeah.
And positive ketones, that strongly suggests ketosis, which could be DKA, but it could also be from starvation or a really low-carb diet.
Right.
And then you have fructosamine.
Right.
That measures those glycated serum proteins. It gives you a shorter-term picture in just the past 2 to 3 weeks.
Right.
And that can be helpful when HbA1c might not be reliable. Like if someone's diabetes management is changing rapidly, or there are big shifts in red blood cell turnover, or they have a specific hemoglobinopathy.
Right.
We always have to consider the factors that can influence interpretation.
Right.
Things like medications.
Right.
Corticosteroids can increase glucose levels. Insulin will lower them. Acute illness, stress, those can raise glucose temporarily. Pregnancy has its own set of guidelines, right?
Right.
And underlying kidney or liver disease, that can make interpretation more complicated.
So it's really about the big picture, isn't it? It's not just the number. It's integrating that result with everything else that's going on.
Absolutely.
Yeah.
It's putting it all together. Now let's talk about the practical stuff. Those special precautions,
Yeah.
the things that we need to do to ensure those results are accurate.
Right.
This is where attention to detail is everything. It starts with patient prep. Fasting for those glucose and lipid profiles, usually 8 to 12 hours. We got to know what meds they're on, if they're sick, if they're stressed. All those things can matter.
Right.
And with the OGTT, that 3 days of carb loading beforehand, that's key too. Patients need to understand that.
Right. And the specimen collection itself, that timing is so important, especially with the multiple draws for an OGTT. We have to follow that order of draw, right?
Absolutely.
And using the right anticoagulant, usually that fluoride oxalate for glucose, that's essential to stop that glycolysis.
Right. And prompt handling is crucial too.
Right.
Glucose levels in whole blood, or if you haven't separated that plasma or serum, they can drop quickly because of glycolysis.
Right.
Ideally, you want to process those samples within 30 minutes. If there's a delay, refrigeration can help,
Mm-hmm.
but don't freeze whole blood.
Right.
That can cause hemolysis.
What about the reagents and instruments?
Same thing, careful handling, maintenance, proper storage, check those expiration dates. No contamination. It's all basic, but so important. And your instruments, they got to be calibrated regularly using traceable standards and maintained properly. And quality control, those programs are essential to make sure those glucose results are accurate.
Right. And sometimes, despite our best efforts, we get those unexpected or questionable results. What are some of the steps that we might take to troubleshoot?
Well, if you see a really high glucose, think about pre-analytical stuff. Did the patient really fast? Was there a delay in processing? Were they stressed?
Right.
And then think about analytical issues. Could be the instruments not calibrated right or the reagents are bad.
Right.
If it's low, maybe the sample sat around too long without that glycolytic inhibitor, or there's bacterial contamination. And if you see weird HbA1c results, think about those interfering hemoglobin variants or method-specific limitations.
Right.
With an abnormal OGTT curve, maybe the patient didn't prep right, or the timing was off, or they didn't get the right amount of glucose. And we always have to be on the lookout for interfering substances.
Right, like ascorbic acid, which can falsely lower those glucose readings with some of those.
Exactly. And you've got other things like bilirubin, high lipids, lipemia, hemoglobin from hemolysis. They could all interfere with spectral photometry. High urea levels in patients with kidney disease, certain drugs, they can also have an impact. And then, of course, those hemoglobin variants for HbA1c, we can't forget those.
Yeah. So it's about being aware of those potential interferences, choosing the right methods, and being cautious when we interpret those results.
Absolutely.
It really highlights how important our role is. Not just running the test, but really understanding all those factors that can affect the results and making sure they're accurate and reliable.
Yeah, you're the detectives of the lab. Now, to tie things together, let's touch on the basic physical and chemical properties of carbs, how they relate to what we do.
Right. Okay.
So the simple sugars, the monosaccharides, disaccharides, they're usually crystalline solids. They dissolve easily in water. They're often sweet. And that's directly related to how they behave in, blood and urine.
Right.
Their solubility makes them easy to transport and measure in solutions. And then their reducing properties, their ability to donate electrons, that was the basis of some of those older tests like Benedict's and Feeling's. Yeah. And it's still relevant to understanding how some of our enzymatic assays work today.
Now, those more complex polysaccharides like glycogen, they're usually not crystalline, they're less soluble, and they're not sweet. That reflects their role as storage molecules.
Exactly.
But it's important to remember that they're made up of those simple sugar units.
Absolutely.
Especially when we think about how they're broken down into glucose, which is what we measure.
Right. And then you have those concepts of cyclization, right?
Right.
Where the linear sugar molecules form rings, and then the formation of glycosidic bonds that link those sugar units together.
Right.
Those are fundamental to understanding carbohydrate structure, how enzymes interact with them in those metabolic pathways, and also in our assays.
Well, this has been an incredibly detailed deep dive into carbohydrates. Looking at it through the lens of clinical lab science, we've covered a lot. From their role as our primary energy source, the intricate pathways that keep everything balanced, to those disruptions that lead to disease, especially diabetes.
Right.
We've talked about the principles behind those key tests, glucose measurements, OGTT, HbA1c, ketone testing. We talked about how important it is to interpret those results accurately, considering the patient's whole clinical picture, and how important that meticulous technique and awareness of those potential interferences are in our work every day.
And I think it's clear that the work you do in the lab, generating those results, absolutely critical for diagnosing, managing, and monitoring a whole range of metabolic disorders.
Right.
You're providing the data that doctors need to make those informed decisions about patient care.
So important. So, thinking about how complex carbohydrate metabolism is and how it really impacts our health, it makes you wonder, what's next for carbohydrate testing in the lab?
Mhm.
Could we see even more sophisticated ways of assessing insulin resistance early on, or new biomarkers to catch those inherited metabolic disorders even before symptoms show up?
It's exciting to think about.
Yeah, it really is. It's a fascinating area to watch. So thank you for joining us for this deep dive. We'll see you next time.